Good to have you here. This is Unintended Consequences, Episode 10. It's May 15th, 2026. Closing the week with one final case study before the weekend. In 1941, a police constable named Albert Alexander lay dying in an Oxford hospital from a simple scratch that had spread infection through his face and bloodstream. Doctors turned to a new experimental drug, penicillin, and watched the infection retreat within days. Yet within a single human lifetime, the same class of medicines that once saved Alexander would help create bacterial strains capable of shrugging off nearly every treatment available, contributing to an estimated 1.27 million deaths worldwide each year. Alexander Fleming had noticed the effect in 1928 while working at St. Mary's Hospital in London. A mold contaminant in a petri dish appeared to kill nearby Staphylococcus. He published the observation, but lacked the chemistry skills to purify and stabilize the active compound. 15 years later, at the height of the Second World War, pathologist Howard Florey and biochemist Ernst Chain at Oxford University assembled a small team to extract and concentrate penicillin. Their goal was straightforward. They wanted to treat the massive bacterial infections that killed more soldiers than bullets in every prior conflict. Early animal tests in 1940 showed dramatic reversal of otherwise fatal streptococcal infections in mice. With wartime supply lines threatened and field hospitals overwhelmed, the researchers saw penicillin as a practical tool to keep wounded men alive long enough for surgery and recovery. They proceeded with the limited information then available. Bacteria appeared to lack the rapid genetic adaptability that viruses demonstrated. The first human trial occurred on 12 February 1941, when Florey's team administered penicillin to Albert Alexander. Although supplies ran out after 5 days and the patient relapsed, the initial improvement convinced military authorities to fund scaled production. By 1943, American pharmaceutical companies using deep tank fermentation techniques produced enough penicillin to treat Allied casualties at Normandy. After the war, civilian physicians quickly adopted the drug for pneumonia, wound infections, and C-Hal birth-related sepsis. In 1951, the U.S. Food and Drug Administration approved antibiotic supplementation in animal feed to promote growth and prevent disease in crowded livestock operations. By the mid-1950s, tetracycline and penicillin were routinely mixed into feed for poultry and pigs across North America and Europe. A 1945 Nobel lecture by Fleming himself noted that there is the danger that the ignorant man may easily underdose himself, and by exposing his microbes to non-lethal quantities of the drug, make them resistant. Yet the warning remained largely theoretical amid visible clinical successes. Bacteria reproduce every 20 minutes under favorable conditions, and each division creates opportunities for random mutations or gene transfer via plasmids. The constant presence of antibiotics in hospitals, homes, and farm environments selected for those rare survivors that could produce beta-lactamase enzymes or alter cell wall targets. Methicillin-resistant Staphylactic acid aureus emerged in a British hospital in 1961, only 2 years after methicillin's introduction, and spread globally through patient transfers and poor hygiene. In livestock, subtherapeutic doses over months created ideal conditions for resistance genes to accumulate. A 1976 outbreak of multi-drug-resistant salmonella in calves traced directly to antibiotic-laced feed on English farms. Third-order effects compounded the problem. Resistant strains traveled on international flights, colonized healthy carriers, and transferred genes to unrelated species such as Klebsiella and Escherichia coli. By the 1990s, vancomycin-resistant enterococci appeared in U.S. hospitals, followed by carbapenem-resistant Enterobacteriacua carrying New Delhi metallo-beta-lactamase genes first identified in 2008. Patients undergoing routine chemotherapy or joint replacements now face infections that require last-line agents or none at all. The same selective pressure that made penicillin revolutionary also guaranteed that its benefits would erode wherever exposure remained widespread and unregulated. Public health agencies responded with stewardship programs that restrict prescriptions and require diagnostic confirmation before antibiotics are dispensed. The European Union banned antibiotic growth promoters in animal feed in 2006, while the United States implemented voluntary guidance in 2017 that reduced agricultural sales by roughly 40% over the following 5 years. Pharmaceutical companies have restarted discovery pipelines, yet only a handful of truly novel classes have reached market since 1980, because development costs exceed projected returns for short-course drugs. Some hospitals now use rapid genomic sequencing to track resistant clones in real time, a partial technological fix that itself generates new data privacy questions. Global estimates still place annual deaths from resistant infections above 1 million, concentrated in low-resource settings where diagnostics and alternatives remain scarce. The original goal of eliminating bacterial disease has been replaced by a permanent management posture. Incentive structures that reward immediate, visible results, whether shorter hospital stays or heavier livestock, tend to externalize long-term evolutionary costs onto future patients and food systems. Complex biological populations respond to uniform pressure by redistributing genetic advantages faster than human institutions can adapt regulations. Decision makers therefore benefit from building in early monitoring for resistance markers and from diversifying interventions rather than relying on any single chemical class. The same pattern appears today in antiviral prescribing during influenza seasons and in the routine use of antifungals on crops. How might current efforts to deploy CRISPR-based antimicrobials or microbiome therapies avoid repeating the same narrow selection dynamic? That's Unintended Consequences for today. If this episode gave you something to think about, share it with a friend who'd appreciate the story. Subscribe wherever you listen, and we'll be back tomorrow with another case. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.